Good morning and welcome to Primary Children's Pediatric Grand Rounds, Pediatric Department Research Spotlight Edition featuring Dr. Martin Tristani-Firouzi on precision medicine. You can ask your questions in the chat anytime along the way in that YouTube live chat, and I will ask those to our speaker at the end. There is about a 10 or 15-second delay on the broadcast, so don't wait till the last second to ask your questions. There are instructions in the chat on how to claim credit today. The code is 4533, and how to watch this later in the day as well. You can claim credit up to 24 hours later. We will also have a slide at the end with the claim credit code. First, I will give the safety story. So a pediatric patient was prescribed amiodarone at twice the daily loading dosage for a 1-month supply with 1 refill, and this was not intentional. There wasn't any documentation on a dosage reduction plan. Upon contacting the prescribing team, the pharmacy called and they changed the dose to the once daily maintenance dose with 1 refill. And so it is a good reminder about stop and resolve if something seems out of the ordinary, and being approachable and open to the pharmacists who definitely have our backs, and we are so grateful for them. Dr. Antonio Cabrera, the Chief of Pediatric Cardiology, will introduce our speaker for this cutting-edge research topic that has the clinical applications as well.
Good morning, everyone. I have the distinct pleasure to introduce Marty Tristani. Dr. Tristani received his MD from the University of Minnesota, where he subsequently completed his residency and fellowship in pediatric cardiology. He joined the Department of Pediatrics at the University of Utah in 1996 as a research instructor and rose to professor by 2011. During his tenure, Dr. Tristani has made a significant amount of contributions in the areas of pediatrics, cardiology, genomics, genetics, and population health. Marty also is a highly respected clinician. He's a productive investigator and engaged mentor. He has received 22 years of continuous extramural federal and foundational funding, and currently has been active in many R01 grants and foundational grants and center grants. Additionally, he's authored more than 85 peer-reviewed publications, and is currently the Associate Director of the Eccles Cardiovascular Research Institute. Specifically, as it relates to this talk, Marty is internationally recognized as an expert in the field of cardiovascular genomics and has successfully translated basic science observations from the lab to the clinic. In particular, the study of precision cardiovascular medicine uses a multidisciplinary approach coupled with cutting-edge research techniques. His lab strives to build the infrastructure supporting personalized medicine by connecting disciplines that excel in genomic analysis, functional analysis, and healthcare information technology while leveraging the unique local resources such as the Utah Population Database. It is for these reasons and many other achievements that Marty has been the recipient of the Edward Clark Endowed Chair in Pediatrics. Marty is going to talk to us today about precision medicine coming to your clinic near you. Marty?
Okay, very good. Thank you for that kind introduction, Antonio. Thank you to Miguel and organizers of the Grand Rounds for giving me the opportunity to speak this morning. A special thanks to my colleagues Lindsay May and Luann Minnick, who helped me out with the last-minute echo this morning, so we're able to keep the child first and always and keep Grand Rounds on time. So we'll start off with I have no disclosures. So since I really created the title of this talk, the talk has evolved a little bit, so please bear with me. But what I'd like to talk about this morning is how we've leveraged electronic health records and genomic data to improve outcomes in healthcare value in cardiovascular space, and leveraging big data to ascertain the historical and geographical distribution of a disease risk allele, and then finally a few comments on where to go next. So I think most people in the audience and at home watching this remember this guy, President Barack Obama. Seems like ages ago, doesn't it? But all the way back in 2015, one of President Obama's accomplishments, many accomplishments, was the creation of a precision medicine initiative. And really what this did is this unleashed hundreds of millions of dollars in NIH research to go into the area of precision medicine, and we were able to leverage those funds almost up to $25 million over the past 8 years or so to create our own version of a precision cardiovascular medicine. So we've heard a lot about precision medicine, but what exactly is precision medicine? So at a very lay level, precision medicine is providing the right treatment to the right patient at the right dose and at the right time. And how do we do this? Well, we do this by combining an individual's genomic profile with clinical information to guide diagnosis, therapy, and intervention. And there are practical applications of precision medicine right now, and that includes the areas of pharmacogenomics, tumor profiling for targeted therapies, polygenic risk scores in adult cardiology are starting to make it into the clinic. But here in pediatrics, we actually have rapid whole genome sequencing in the NICU, which is really at the forefront of precision medicine. Right now, we have these programs occurring at the University of Utah Newborn Intensive Care Unit and Primary Children's NICU. This is under a research umbrella, but I believe in the next 2 years, this will be standard of care across the country. So this is a white paper that was published recently in October 2020 in the journal Nature, which really describes a strategic vision for improving human health at the forefront of genomics. And so basically, what the Precision Medicine Initiative Obama did is it released money that focused in this area right here. That's basic genomic research. And this is really the space that we've occupied for the past 8 years or so with genomic technology innovation, genomic data collection as far as large cohorts of congenital heart disease patients, arrhythmia, and sudden death victims, as well as novel and innovative genomic analytical tools. So we've done a really good job on the basic genomics research side of things. But we have done less or made less progress here on what is called the genomic learning healthcare system. And this is really the application of all this basic genomic research onto the clinical side of things. Now, we do have some grants in the genomic medicine practice innovation space, and I'll talk about that. But this is really the challenge for the next 5 to 10 years for all medical centers, including Primary Children's Medical Center, including the University of Utah, including Intermountain Healthcare. How can we bring these basic genomic research findings into the healthcare system so that precision medicine can become a reality? So why isn't it a reality right now? Well, it's not a reality because it's very hard. I mean, there's huge amounts of data. In fact, we're drowning in a sea of data. So understanding the contribution of genetic variants to a patient's risk for disease remains extraordinarily challenging, and computing on millions of electronic health records is a daunting challenge as well. So what I'd like to present in the next few slides is my vision for what I see as our Utah solution for genomic healthcare learning system. And that's leveraging existing infrastructure and resources such as the Utah Genome Project, which has sequenced the exomes and genomes of thousands of Utahns, the Utah Center for Genomic Discovery and my collaborators there, who really work on the bioinformatics side of things, and of course the Utah Population Database, which is the largest genealogical database in the world that's linked to medical records. One of the things we're working on is what we call a point-of-care genomic analysis tool. And this is a project together with my colleague, Dr. Gabor Marth, who's the co-director of the Utah Center for Genomic Discovery. And we're able to leverage an R01 from a program called the Clinical Sequencing Evidence Generating Research Program, or the CSER program. And this was really to design a web-based tool for physician-driven diagnostic interpretation of data. And the idea is to make a web-based system that is accessible to clinicians, easy to use, and intuitive. And basically what we've done is we've designed sort of a step-by-step process that the physician would walk through in analyzing the exome or the genome, going through some basic quality control, choosing genes that are relevant to the phenotype, and looking at variants in those genes, and finally coming up with a conclusion about whether any of those variants might explain the phenotype. And unlike Icentra, which I think many of us think never actually had any clinician input ever, we were engaging our clinician stakeholders very early in the game, and then repeatedly. So we would bring the clinicians together, sit them down, walk through the tool, and get information about what worked, what didn't work. And you can see in this photo here, we've got the heads of state of precision medicine here at Primary. We've got Josh Bonkowski, Dave Viscaccio, that's Lorenzo Botto back there, Russ Butterfield, Susan Etheridge, and we have some of the diagnosticians from ARUP as well. So the idea is to get input directly from people who would use this and find ways that we could integrate this into clinical care. So that's ongoing. What we've done more recently is we've adapted that same system, the general model and the general sort of bioinformatic and software infrastructure, to address a recent call for applications from the NIH regarding improving genetic counseling process. So this was a grant we submitted in October, and we'll find out our score in February or so. But what's great about this is it brings in multiple stakeholders into the decision-making process: the genetic counselors, the clinicians, and the molecular diagnosticians, all together in a space where people can communicate back and forth and help to adjudicate variants. And again, it's the same general idea of walking through a very structured process. And this is something that we actually do on a regular basis. In fact, we had our inherited arrhythmia clinic on Tuesday, and basically walk through variants with our genetic counselors, with our clinicians. And it's really important because the genetic testing companies and the molecular diagnosticians are extraordinarily conservative. So they're not going to call something likely pathogenic unless there has already been someone who has been reported to have that variant or there's existing functional evidence. And yet a clinician can say, you know what, I know this patient has this clinical diagnosis, and this is the gene that is responsible for that diagnosis. And this is a very rare variant, never reported. It's highly conserved. It's predicted by multiple tools to be damaging. And I think it actually will explain the disease in the patient. So as a team, you can arrive at that kind of a conclusion. But as an individual, it's very difficult to do that. So we're hoping that we're going to be successful in this grant and we can move this forward into our inherited arrhythmia clinics and some of the other clinics across pediatrics. The other interesting thing about this system is we've designed a patient portal system that would allow the patient and the family themselves to investigate what is the variant that's under consideration with some simple things about what's a recessive disorder, what's a dominant disorder, what does this variant mean, and there's an area over here for them to give feedback about whether they understand this or don't understand this, and then this can help the genetic counselor as they go through the counseling process. So one of the things that we've discovered as being part of a consortium of centers studying the genomics of congenital heart disease is that isolated congenital heart disease is actually very uncommon that you will find a genetic explanation for it. So the majority of cases, we don't find anything, even with whole genome sequencing. And so what we wanted to do is find a way that we could still provide precision medicine to these sorts of patients, moving beyond genomics in precision medicine and taking advantage of big data analytics. And so this is another really hot area of research in the space of big data analytics. So there's a lot of promise and a lot of potential, but of course, there's a lot of challenges in making this work in a practical way. So the question then is, how can we harness the power of big data for discovery and outcomes research? So what I'd like to do in the next few slides is introduce our methodology, which is currently under review at Nature Medicine. We call this technique the Poisson binomial-based comorbidity discovery tool, or PBC for short, so I'll just stick to PBC if that's okay. And the goal here is to discover comorbidities in the electronic healthcare data. And by comorbidities, what we mean are not just diagnoses, but procedures and medications that co-occur together greater than expected by chance, and to identify temporal dependencies, which disease comes first, in that process and quantify the transition rates between those comorbidities. And really the engine, the analytical engine behind this is my colleague in human genetics, Mark Endowed, who's also the co-director of the Utah Center for Genomic Discovery. And basically what we use is we use the electronic data warehouse at the University of Utah, which is really a mirror of the electronic health record that we use on a regular basis at the U, but it allows for computation, so we're not slowing down clinical care. And every night the EDW is updated. And of course, the Utah Population Database. So just looking at some numbers, the UPDB has almost 14 million individuals in it. And the EDW has about 3.7 million individuals. And when we intersect them together, that gives us close to 2 million individuals that are part of the Utah EDW and part of the UPDB. And when we go through and sort of curate that data a little bit, we get down to close to 1.6 million people who are available for computation. All right, so if we're to consider two diagnoses, atrial fibrillation and stroke, these are things which are known to co-occur. So if you have a diagnosis of atrial fibrillation, you're at increased risk of stroke. So we want to look at how can we prove these sorts of relationships between different diagnoses at scale, looking at hundreds of millions of records, okay? So typically, to adjust for demographic variables, most methods would stratify the population by age and sex, and then calculate statistics such as a chi-square for each stratum. So what we do differently is we use a per patient logistic regression, and then we use the Poisson binomial distribution to calculate the p-values for the association between terms. And so when we look at various demographic records and their influence on medical terms, what we find is that sex, either male or female, influences about 40% of all medical terms in the EDW. Ancestry, meaning Caucasian, African ancestry, or Asian, influences about 27%, ethnicity 37%. What kind of insurance you have influences 30% of the medical record terms. And something that we call EHR exposure, which is how many visits you have in the EHR, how old you are, every medical term is influenced to some degree by that. And so basically what is done for every single patient and for every single diagnosis, medication, or procedure, a linear regression is performed on that individual patient, and then that's summed up across all individuals in the electronic health record. So this is kind of a complex slide here, but I just want to walk through to show the power of this method to detect relationships and why the stratification method tends to fail. So we believe that our method retains power as we add features, while stratification lowers the strength of p-values as a function of added features and as a function of the size of the stratum. So let's just sort of look here. What we've got, again, is our diagnoses, atrial fibrillation and stroke. We have the p-values for the PBC method and the stratification method. We're going to start off with 1.5 million records in both cases. And if we just look across all those 1.5 million records, the p-value for the association of atrial fibrillation and stroke is 1 times 10 to the minus 31,000. So extraordinarily strong, right? But you would say, yeah, but there's so many things we need to account for: sex, age, et cetera, et cetera. So when we add female sex, the p-value drops. It's still super significant, right? 10 to the minus 16,000. But then when we say, well, let's look at women who are age 50 to 59, our p-value is starting to drop. When we add in ancestry, whether they're African-American ancestry, our p-values drop. Adding ethnicity further, when we say what kind of health insurance, now we're outside of the statistical range for a relationship. By contrast, the PBC method, the p-values are retained as highly significant across all of those based on the type of linear regression model system that we use. So what happens if we start with smaller number of electronic records? So in this case, we've taken from those 1.5 million records, we've randomly selected out 95,000 of those records, and you can see that as we stratify that down based on female sex, age 50 to 59, and ancestry, now we've lost that ability to detect the difference. whereas with our PBC method, we retain this ability to find the significant values. So that's sort of our starting corpus. What are the statistical relationships between medications, diagnoses, ICD-9 and 10 diagnoses, and procedures or codes coded by physicians? What's shown on this map here, which we call a patient disease network, is a total of 50 million relationships, 50 million comorbidities discovered in the entire EDW at the University of Utah, encompassing 40,000 diagnostic codes, over 5,000 procedure codes, almost 2,000 medications. And the important thing here is at this point, there's no PHI. There's no patient per se that's here. It's just the relationships between diagnoses, medications, et cetera, et cetera. And each little dot on this network here is a single disease diagnosis or procedure. And they cluster together based on how related they are. And so what you can see in this cluster here with a lot of cardiovascular diagnoses, we have atrial fibrillation, we have hypertension, congestive heart failure, et cetera, et cetera. That's a blow up here. And then when we look out further across the entire disease network, we can see other diagnoses and medications scattered across that I've highlighted. And what we're going to focus on in the next couple of slides is the diagnosis of heart transplant. So each of these diagnoses can be related to other diagnoses by following the track. And given that there's 50 million of them, it's really hard to follow that, right? But we know those connections exist, and we can represent those connections in what we call a term trajectory for heart transplant. So here's our diagnosis of heart transplant, and we can calculate the transition rates and the temporal directionality of diagnoses. So in the EHR, in the EDW at the University of Utah, a patient with a diagnosis of ischemic heart disease has about a 19% chance of ultimately developing a diagnosis of heart failure. If you have a diagnosis of heart failure, 3.5% of those individuals go on to get a diagnosis of cardiogenic shock. 32% of those go on to require milrinone. 5.5% of those go on to get a heart transplant. And a third of those that have a heart transplant get a diagnosis of an infectious disease. So this procedure, this PBC, really allows us to create these connections, quantify the directionality of them and the transition rates. But what it doesn't tell us is if you have ischemic heart disease and heart failure, what's your chance of getting a heart transplant? And so that requires a different methodology, which I'm going to talk about in the next few slides. So our PBC approach is powerful, but it only describes relationships between two variables. So this is the patient disease network that for pediatric heart transplant, and this is using data from the University of Utah EDW and the Primary Children's Echo database. So that's sort of our starting corpus of records. We apply our PBC methodology, and again, we get this patient disease network. It looks a little different because the information going into it is a little different. But here's a diagnosis of transplant, and you can see it's connected to a whole host of other diagnoses scattered throughout the disease network. But it allows us then to calculate the risk for a heart transplant if you have a particular diagnosis. And so what I'm going to focus on here is having a diagnosis of hypoplastic left heart syndrome. So the absolute risk for all our patients with HLHS is about a 13 or almost a 14% chance of requiring a heart transplant in their lifetime. And that's an 83-fold increase over the baseline. All right. But again, this shows that we can capture relationships between two variables, but not three variables. So some patients with hypoplastic left heart syndrome actually go straight to a transplant. All right, not many here, but we do have some that do. Others go on to get a palliative procedure called the Nora and then later go on to transplant. So if we want to understand the risk of transplant, if you have HLHS and you've had a Nora, we need a different technology to do that. So again, the PBC method allows us to identify comorbid variables, but we need an artificial intelligence solution to solve the multimorbid calculations. And that artificial intelligence solution is called Bayesian networks. So you probably never thought that we'd be talking about Bayesian networks on pediatric grand rounds, and I agree with you on that, but just bear with me on this. So just a little introduction about what Bayesian networks are and the advantages of them. They allow us to combine demographic, clinical, and genomic variables. They allow us to investigate interdependencies and effect sizes. They handle missing and unknown values correctly. And really importantly, they can capture and measure non-additive or synergistic impacts, and we're going to look at that. Very importantly for nowadays, especially in healthcare, this is an explainable form of artificial intelligence. You can do the same thing if you use neural networks, but neural networks are what's called black box algorithms, where you don't know why the computer came up with that value. With the Bayesian network, you can find out exactly how you came up with that risk calculation. And again, the important thing is at this stage, no PHI is involved. So let's walk through an example of how we can apply the Bayesian network to risk predictions within children with congenital heart disease. So if we want to know what is the probability of observing a patient in our database with a bicuspid aortic valve, that value is about 4.6%. And if you would say, you know, I thought that a bicuspid aortic valve was more like 1% of the population or maybe up to 3%, you'd be right. Our value is a little higher than that, but remember our starting corpus is a child that's had an echo. And so that's not the same as the general population. So in our database of children who've had an echo, 4.6% of those have a bicuspid aortic valve, close to the value in the general population. So then if we want to know what's the probability of observing a male patient with a bicuspid aortic valve, you might think, well, all you have to do is take the probability of having a bicuspid valve and multiply that by the probability of being male. Well, it turns out that that's not true. That won't give you the right answer. And the reason is that sex and bicuspid aortic valve are conditionally dependent variables. And the Bayesian network is able to capture that information. If we were, the value of observing a male patient with a BAV is 6%. It's higher than the background because of this conditional dependency. If we were to just multiply those two variables together, we'd get the wrong answer. So it's well known that males have an increased risk for bicuspid aortic valve, and our data tell us that if you're a male, you're at 2.2-fold more likely to have a bicuspid aortic valve compared to a female, and that's consistent again with what's been reported. So what we're trying to show in this talk here and in our initial paper is just kind of recapitulating things that are known so that we can show that our methodology is capturing things correctly. So one other important thing is, as cardiologists, we know that sex and coarctation synergize to increase the risk for bicuspid aortic valve. And again, the Bayesian network is able to capture these two conditional dependencies. So the probability of being male and having a coarct increases your risk of bicuspid aortic valve almost tenfold compared to being male and not having a coarctation. Again, a gut check for data which we already know. So the basic workflow here is we start off with some corpus of data, whether it's the EDW at the U, whether it's primary children's EDW, whether it is the ECHO database, whatever clinical database. We run our PBC approach, we generate our patient disease network, and then we run Bayesian networks to do our multi-morbid outcome calculators. So let's go back to our example earlier about adult transplant. So over here, this was what the PBC approach is telling us. When we run our Bayesian network analyses, we get a different looking network, okay? But here we have heart transplant with its connections to those multiple variables. But what that allows us to do then is to calculate the combination of risks. So a patient at the university who is on milrinone has a 5.4 risk of requiring a heart transplant, absolute risk. Based on the data, that's a 175-fold increased risk over baseline. If you have cardiomyopathy, you have an 86-fold risk over baseline. Well, what about if you have both milrinone and cardiomyopathy? Well, you can see these are not additive. This is a 407-fold increased risk. It's not the same as just adding these together. So again, the Bayesian network has captured that non-additive and synergistic effect between variables that will then allow us to make the proper risk predictions for individuals. So what I'd like to show next is how we can integrate genomics into these Bayesian networks. And what we're looking at here is data from about a little over 3,000 probands, or individuals with congenital heart disease who are a part of the Pediatric Cardiac Genomics Consortium, or PCGC. Almost 1,000 of those individuals have whole exome sequencing. And what the variables we're looking at here is we're focusing on the risk of death. And the variables that are brought into the Bayesian network include the phenotype, which will be hypoplastic left heart, heterotaxy, conotruncal malformations, which include tetralogy of Fallot and transposition, LVOT, or left ventricular outflow tract disorders. Those are primarily coarctation, and ASDs. What I did here is I collapsed all the postoperative complications into a single variable just for ease of the compute. These complications include things like arrhythmia, stroke, thrombosis, pleural effusions, and the need for ECMO. And then we have other variables which include the patient's sex, prematurity, and we have two different types of gene lists here. One is called a MIPS gene list, and we'll just say this is a gene list which has been sort of selected by the PCGC as containing genes with a high likelihood of having related to congenital heart disease. So this would be damaging genes in the MIPS list. So what happens when we look at the risk of death based on whether a patient has hypoplastic left heart syndrome or a damaging gene? So if you have a damaged gene which is related to congenital heart disease, that increases your risk 3.3-fold, let's just say, over baseline. If you have HLHS, the risk of death is almost 5-fold above baseline. And yet if you have a damaged gene and you have HLHS, again, 11-fold increased risk of death. So it's a non-additive effect, which we have discovered with the Bayesian network. We can also use the Bayesian networks to look at the impact of maternal health on the health of the child or the fetus. So in this Bayesian network, what we have is a maternal diagnosis of pregnancy-induced hypertension and the risk of having different congenital anomalies. And basically what we find is that if a mother has a diagnosis of hypertension complicating pregnancy, that there's a 1.7-fold increased risk of having a child with a cardiac anomaly. All right. Now, again, these multimorbid or multiple calculations allow us to say that if the mother has hypertension complicating pregnancy and the child has heart disease, well, they're at an 11-fold increased risk of having a nervous system disorder or a neurological disorder. So again, this allows us to peer into the medical records system and pull out relationships that can help us in predicting outcomes of our children. So where do we go next with this sort of general type of technology? So I think the really exciting thing here is that with philanthropy, Intermountain and Primary Children's have announced this Precision Medicine Center, which is funding rapid whole genome sequencing in the NICU, among other projects. The University of Utah has its NICU rapid genome sequencing, and I think that this really is going to be the standard of care for the future. And what I envision then is that we need to find a way to incorporate genomics into our weekly rounds in a way that, that can help overall with quality of care of these patients. And again, that gets back to that idea of the genomic learning health system and how can we do that. So based on our experience with the University of Utah EDW, I predict that we can estimate individual risk for any medical disorder within Primary Children's EDW. That is, then we can say what's the risk of childhood obesity based on any number of factors, diabetes, seizures, you name it, anything which really affects us in the types of patients that we take care of, we will be able to calculate individual risks for those patients. But what's missing here? Well, the absolute requirement for this to become a reality is we need access to the entire contents of the Primary Children's Hospital EDW. So this is something which is a huge hurdle, but it's something that we have to overcome collaboratively if we want to make Primary Children's on the forefront of precision medicine. We already have permission and access to University of Utah EDW. That helps us because we're all university, or most of us are University of Utah employees, and all of our coding goes through the University of Utah. So there's a footprint of our patients in the University of Utah EDW, but the rich data is in PCH. So we need to find a way to overcome those hurdles. The other thing we need is we need access to the raw genomic data. So currently, the rapid NICU sequencing done here at Primary Children's is performed at Rady Children's in San Diego. They control the genomes. The plan is that that will be taken over by Intermountain in the future. But again, access to the raw data is going to be crucial if we want to use these tools cutting edge-wise to make Primary Children's at the top of precision medicine institutes. So in the last portion of the talk, I'd like to present our recent collaboration between the University of Utah, Intermountain Cardiology, and Ancestry DNA. And this is just a really cool and exciting story that spans genealogy in Utah and the movement of people across continents. And it can really only happen as a result of a collaboration. So we have a manuscript which is currently under review at Nature Communications, titled The History and Geographic Distribution of a KCNQ1 Atrial Fibrillation Risk Allele. And so atrial fibrillation is the most common sustained arrhythmia in clinical practice. AF patients have an increased risk of stroke and early cardiovascular death. There's a huge economic impact of atrial fibrillation, over $7 billion in annual healthcare costs using data from like 5 years ago. And the interesting thing is that young onset, or what's called lone atrial fibrillation, has a very strong genetic component. Here we can see our normal sinus rhythm with our P, Q, R, S, and T, and you can see these fibrillatory waves here consistent with atrial fibrillation. So together with our partners at Intermountain Healthcare, University of Utah, we actually put together a pedigree spanning 8 generations with a family of autosomal dominant phenotype of young onset atrial fibrillation. And the story is just super interesting. So there was a family over here that was seen by Dr. Steve Blyle, one of our geneticists years ago, probably 12 years ago, for a newborn that had a congenital disorder. And the mother said to Dr. Blyle, you know, this is all interesting and stuff, but we have a lot of people in our family with atrial fibrillation. Can you help us figure out why that is? And so Steve emailed me years ago and said, hey, I've got this family with young onset AF. What do you think? And I'm like, God, I'd love to study them. We just don't have the resources. We don't have the money. We don't have the grants. So as time went on and we started to get access to some seed money from the Utah Genome Project, I remembered that patient and was like, who was that patient? Or I remember the interaction. So I asked Steve Blyle, he went through his old emails, I went through my archived emails, we found the original email that had that description, we found the mother, we reached out, and we ascertained this side of the family here, and separately from Intermountain Cardiology, they ascertained this side, then we came across this side of the family, and before we knew it, we had realized that all of these individuals, without even knowing it, were related to each other, related to individuals that came to Utah, were born in the early 1800s, but came in the 1840s to settle in Utah. So basically what we're showing here is, in red, a diagnosis of AF onset younger than 60, so you can see all the red there coming across, and an AF onset greater than 60 is in dark blue. And one of the features here is you can see that individuals that have that dark blue, that have an AF onset greater than 60, don't have any children who go on to develop young onset AF. So in all the cases of blue, you don't see transmission. But in all the cases of red, the young onset, they have children that have young onset AF. We found that another phenotype associated with the family is fetal bradycardia, and these children over here, some of them were actually delivered premature because of concern for fetal bradycardia, not knowing that they carry this mutation. Then the other sort of interesting thing is there's 2 members that actually have a clinical diagnosis of long QT syndrome. One young girl here presented with atrial fibrillation at 13 years of age, and also had long QT syndrome. So this branch of the family we were following clinically in our inherited arrhythmia clinic anyways. But it was remarkable how with the Utah Population Data Resources, we were able to create this connections between all these individuals. And so looking at the characteristics of those that had the younger onset, younger than 60, the average age of onset is really 32, which is extraordinarily young for AF. There were 10 individuals that had the AF onset greater than 60, and these were the more typical age-related phenomenon of atrial fibrillation. You'd expect to see that in a pedigree of this size. The total number of patients that we genotyped as positive were 36 that carried the mutation, and I'll talk about that in a second. 2 with lung QT, 1 sudden death, and 11 with fetal bradycardia. So when we look at the pedigree, we really can only show you who's positive because there's too many patients. But the power, again, of the Utah Population Database is we know who all those other individuals are. And so what this graph here is showing a circular pedigree. So here are the founders. Here's the first generation, second, third, fourth, et cetera, et cetera. And what we're showing here is the transmission of young onset AF across generations. And this does get back to the idea about precision medicine. So if you look at these individuals here, there's hundreds of individuals which you would never have to worry about that are going to have young onset AF because they're not in the pathway of having of possibly even having it. It's really these pathways are the ones that require screening of the children and notification to say, hey, if you have palpitations, don't ignore it because you might be in atrial fibrillation. So what is the gene that's defective that causes this? This is a plot that includes all genetic variants across all the chromosomes in 5 individuals for which we did whole exome sequencing. And the top hit here is a gene called casein Q1, which is an ion channel, and the p-value reaches Bonferroni correction for significance. What we can see here are genes very close to casein Q1. We call this kind of a comet tail, and these show up because they're in linkage disequilibrium with the variant of interest. So this casein Q1 is an ion channel. Mutations in this ion channel are known to cause long QT syndrome, but gain of function mutations are known to cause atrial fibrillation. So our model organisms for model organism systems for studying genetics include the zebrafish and human-induced pluripotent stem cell-derived cardiomyocytes. I'm just going to present some of the data on iPS cardiomyocytes, but I think most people in the audience know that the Nobel Prize for Physiology back in 2012 went to 2 individuals who really designed the mechanism to turn somatic cells into stem cells, and it really comes down to 4 genes that, when turned on, called the Yamanaka factors, will turn on the pathway for pluripotency. And we can either use fibroblasts or peripheral blood mononuclear cells. We use peripheral blood mononuclear cells because they're really easy to get. You don't need a skin biopsy. We transfect those cells with a virus that carries these 4 Yamanaka factors, and it turns them into iPS cells, which then differentiate into beating myocytes. And it looks like my movie isn't playing there, but that would show the beating myocytes in a dish, which is just an incredible sight. Every time I see that, it's like amazing that these cells have differentiated into heart cells and they beat in a dish. And we've designed a system for moderate throughput analysis where we can measure action potentials not with a pipette, but instead with a voltage sensitive dye, which increases our throughput dramatically. So when we look at stem cell derived cardiomyocytes from 2 patients that are healthy, one of them is a sibling of this patient right here, and we look at 2 patients that have are heterozygous carriers of the mutation, what we see is that the duration of the action potential here, APD50, is shorter when we look at all the cells, and this is about 200 some cells per group that we looked at. They're much, much shorter. When we look at cells that we believe are destined to become atrial cells and those that are destined to become ventricular cells, both the atrial-like and the ventricular-like action potentials are shorter. So this then provides the substrate for atrial fibrillation, because if you have a short action potential, you're able to repolarize the atrium and sustain the extraordinarily fast rates of atrial fibrillation, where the atria are beating 300 to 600 beats per minute. So we partnered with Ancestry DNA in order to get information about how old this genetic variant is, where did this variant come from, and look a little bit about sort of the global history and geography of this variant. And this is a methodology that was published by Ancestry DNA in a manuscript in Nature Communications in 2017. And it's a little complicated, but let me just walk through the methodology because it has relevance to what we want to do with our variant. So basically, what Ancestry does is it looks across the genomic sequence of individuals, and it says how related are those individuals. And they do that by analyzing what's called identity by descent, or IBD. So the closer you are, the more related you are, you share more genetic material. And so they can create these networks of individuals that are related, and then they detect those network clusters by saying, all right, this individual really belongs to this cluster, this individual belongs to that cluster. And they further analyze that using principal component analysis to say, this is a distinct cluster, this is a distinct cluster, and this is a distinct cluster, okay? So then what they do is they say, all right, now what they're going to do is they're going to look at individuals within those specific clusters and look at sort of their genetic makeup and look at where their ancestors were born. Okay, and this is something that really only Ancestry DNA can do because they have all those records and all those genealogies from their subscribers. So let's look, this is a cluster for African Americans. Let's look at this individual right here. So what you see here is about 75% of their DNA maps to regions of Africa listed here, Ghana, Cameroon, et cetera, and about a quarter of their DNA maps to Northern Europe, Scandinavia, Great Britain. So this is an African American, has 75% of their DNA with origins in Africa. So then they say, okay, this person actually has a genealogical tree and ancestry. They were born in Los Angeles, their parents in Virginia, their grandparents in Virginia, their great-grandparent in Pennsylvania, and a great-grandparent in London, all right? That's how they get this Great Britain and Scandinavian DNA, right? So what they do then is they say, okay, they do this across all the individuals in that network, and what they're showing over here is the ancestral birth locations of those individuals. And these are odds ratios here, so in red we have very high odd ratios, greater than 10, of the probability of that individual having a relative that was born in the Deep South. So this is the distribution of all the African Americans in the Ancestry DNA database and where their relatives were born 3 generations ago, which would make sense as the majority of these individuals were born in the Deep South. So one more example before we go on to our research. So obviously people know that French Canadians, Cajuns, and Acadians are all related to France in some way, and this map is actually highlighting that. So the cluster of French Canadians, Cajuns, and Acadians, they all cluster together because they share DNA, right? When they look back to 1650 to 1790, they find that the majority of those individuals are born in France, but at the same time, a fair number of individuals have already been born in Quebec, Nova Scotia, and Louisiana. When they look at a different time period, here is 1785 to 1878, you can see that most of the relatives are no longer being born in France, rather they're being born in these 3 distinct regions of North America. When they look 2 generations ago, almost no one born in has an ancestor born because everyone has already moved and relocated to North America. So we're going to apply the similar technology to our family here with this mutation in the KCNQ1 gene. And so what we did is we took 5 members of that pedigree, we recruited them for a sub-study with Ancestry DNA. Here are the 5 individuals, and what this map is showing is the red color means 2 meioses away. Okay, so this would be, we know this is actually an aunt, but it could be an aunt, an uncle, or a grandparent. This is 3 meioses away, this is a first cousin. And this is a person who's actually 11 meioses away from this individual. So 11 meioses away is a very, very far distant relative. And in fact, it's almost the same distance as if you took two people in the United States of Northern European descent. So while these individuals are all part of the same family, some of them are extraordinarily distant, but the tiny little piece that they happen to inherit is the piece that carries the mutation. All right, so just in the interest of time, we're going to kind of skip this slide over here, but this is the methodology that we use to ascertain who are the relatives of the 5 Utah individuals. So again, using that same methodology, which Ancestry DNA's commercial market is based on, these are all individuals that are related to person C. These are all individuals that are related to person D. And here in the center, we have individuals in the Ancestry DNA database that actually are related to all 5 of these people. And these are people that we believe would actually carry the mutation. These are individuals that are related, but likely don't carry the mutation. These are individuals that are related and likely carry the mutation. Now, Ancestry has strict policies over who can be sequenced, and only 3 of these individuals here in the center had given permission to be sequenced, but all 3 indeed carried the mutation. All right, so now applying that same methodology that we talked about in the previous slide, we're looking at the birth locations of individuals. When we go back to 1700 to 1800, we can see that the location of individuals, those 5 individuals, those relatives, they are centered right here in Denmark. You see a few people here on the northeastern border. When we look at the time period 1800 to 1850, you can see that distribution is now decreasing from Europe and showing up on the eastern border. But not only that, we're showing relatives that are being born along what is basically the Mormon migration trail coming out to Utah. And then when we look at the 1850 to 1910 time period, you can see this high localization of those relatives now being born in Utah, with just a handful still being born in Denmark. So again, this is telling us these odd ratios are extraordinarily high. This is 2 to the 8th power, 2 to the 9th power. So the statistics are telling us what basically, in a sense, the family has believed all along, that their relatives came from Denmark, and these individuals, actually, the founders, were born in Pennsylvania. So again, this allows us to look at the migration of this mutation, or individuals who carry this mutation, across time and across continents. And just for clarification, this map here is showing the relatives of those 5 that carry the mutation. So when we look at that smaller group of individuals that actually carry the mutation, or that we believe carry the mutation, we see a similar process going on. It's just that we don't have enough individuals. We don't have the same power to show a map like this, but the process looks very similar. So the next question we wanted to ask is, how old is this mutation? When did this mutation come in to the human population? And this is sort of the expertise of the individuals, our colleagues at Ancestry DNA, and they used a technique called GEVA, or genealogical estimation of variant age. So I won't even begin to go into the math related to that because I don't get it. But basically, the standard line is they use a hidden Markov model to identify the length of inherited identity by descent, or IBD, sharing around the mutation between pairs of individuals, and then they estimate the time of the most common recent ancestor. So this is a histogram here showing all the variants that are unique to 2 mutation carriers from Utah using whole genome sequencing. And what is listed down here are generations before present. And so there's a cluster of variants which are very, very old. 1,000 generations, we assume a generation is about 25 years, so that's 25,000 years old here. And the variant that, the mutation actually is among the youngest of all the variants that are on this, that are carried by these 2 individuals. So the estimation is that the mutation came into the human population around 182 generations ago. There's a very broad confidence interval, 80 to 720 generations. But if we assume that there's 25 years per generation, that would mean that this mutation entered the human population in around the Bronze Age. And the idea here is that this is a mutation which may not affect survival or reproduction, actually, because it does happen in young adulthood, but it certainly can impact reproduction. If an individual is 18 years old and born in the Bronze Age and has atrial fibrillation, they can't just go and get cardioverted, right? They're definitely going to be impacted and be unable to really participate in life in a significant way, and will likely end up with a stroke. So the idea is that there was probably some impact on fitness, but because in many cases individuals would have already had children, this is a way that this mutation was able to propagate over time. And if we just sort of look in comparison, this is the mutation here, and this is the mean age of all variants that are on casein Q1 when you look in the 1,000 genomes data set. And you can see that some of these variants are extraordinarily old. So these are variants that occurred, almost 500,000 years ago, so very, very old in evolution. But those cannot be variants that cause disease. The disease-causing variants are always very young because those tend to be selected out over time. So just in the closing minutes here, I just want to close with the idea of my optimism that we will be able to indeed create a genomic learning healthcare system here at Primary Children's, that we will be able to leverage the big data existing in the electronic health records and genomic data with our final goal of improving outcomes and improving healthcare value. So I think the next coming years are going to be extraordinarily exciting with the advent of whole genome sequencing, both in the NICU and in the other ICUs. And just like to thank a lot of people, members of my lab, members of the Utah Center for Genomic Discovery, Primary Children's, Intermountain Healthcare. These are obviously experiments, and these are grants that require a tremendous amount of collaborative effort. And that's really just the way forward. It's all about collaboration, forming partners that share the same goal of really ultimately getting precision medicine into the clinics and having an impact on patient and patient care. So thank you very much, and I'm happy to take questions.
Excellent. Thank you so much, Dr. Tristani-Firouzi. Thanks for teaching us. It seems like now that the genome sequencing and gene identification is kind of easier, the tools for interpreting that ocean of data. I mean, you just think about back when you learned how to interpret a blood gas, and even just that. I mean, you could, oh, now it's just 30 million base pair exome I have to interpret. And I could just imagine people coming in with that direct to consumer, at the pediatrician office, and what do I do with this? So but having these kind of tools coming down, I think that's so important. So let's open this up to the questions that have been asked in the chat. Dr. Rashmi Shah answered one of them already in the chat, so that was really nice. Dr. Bonkowski asks, he says, great presentation. One question that you and all of us in this space wrestle with is the balance of advancing health and discovery and potential privacy concerns, especially with the large health and genomic data sets. What do you see the field doing with this challenge over the next 5 years?
Yeah, that's really a great question. So we have to start off with PHI because we have to know which patient is which. But once we do that calculation for the relationships and the comorbidities, at that point, there is no PHI because the patient has been removed from that. So that's a data set which anyone can compute on, which anyone can look at individual risks. And so we do need PHI to get there, and we need PHI at scale, the entire EDW. That's really what we're talking about here. So that, I think, is very scary for administrators, like, well, you can't do that. But we have found mechanisms of doing that, right? We're computing on the Utah Population Database. It's one of the most secure servers in the state. So the EDW, same thing, highly secure server. We're computing on that, and as is other people as well, right? And so within the IT system, there are multiple systems of safety to prevent access or unauthorized access to those. And I think that we can apply those methodologies to get the data in a usable format that no longer relies on PHI.
Right, and you always want to have a cybersecurity team, that these big institutions have. Um, with such tiny p-values, it seems like 10 to the minus 10th narrows it to one person on Earth, right? But is there a chance this is overfitted genetically to Utah? Ashwin Lal, Dr. Lal
Mm.
asks, will these techniques be helpful with patients without the extensive family roots in
Yeah,
Utah?
yeah. So actually, another really great question. So the so obviously this data that we presented is unique to Utah because it's based on the Utah data. And there are unique features about our geography and our ancestry and patients here and how they present. And part of our renewal for this pediatric cardiac genomics consortium is to create similar databases at the other centers by using data pulled out of what's called a network of networks, CoreNet, where data is stored in a common data model. And our goal is really to leverage those existing data sets at those centers. Again, to do these computes, initially you have to have PHI, and that would all be done at the local center. But once it's stripped of the PHI, then that raw data can be compared between institutions and then combined to get risk factors for the general U.S. population.
Yeah, you get kind of more clusters and nodes that way. Well, thank you very much. The time is up for this session. The Cloud CME code is 4533. And now we're going to have the time where, you can leave if you need to, but maybe we could pretend that we're coming up to ask Dr. Tristani things afterward. Would that be okay?
Yeah.
Do you have a minute? Okay. Well, thanks everybody for coming. And this concludes the session. We'll just we'll keep the audio on the broadcast for a minute. So another question is that we noticed that getting granular with the genomes and the ancestral birth locations, it seems like that has actual biological meaning, unlike the social and political constructs like what race somebody is, a kind of a made up thing. Whereas the actual country of somebody's ancestral origins maybe actually have some genetic meaning. So the more transparent and explainable Bayesian network, do you think that is one way to protect against the phenomenon where we've seen AI, like artificial intelligence, amplifying racial health disparities? Like when we've seen with some of the decision algorithms that AI comes up with, and they feed race back into it, and it makes the racism worse. Kind of like the more transparent things where it's like, these are cardiac things. Is that one way you protect against that?
Yeah, that, again, really insightful question. So what we use out of the EDW is reported ancestry, reported ethnicity. Once we have access to genomic data, we don't have to rely on the report because the genetics will tell us what is the admixture, what's the degree of admixture. And so that will allow us to more precisely calculate the contribution of risk, for example, risk of hypertension with an African American male at a specific age. So the data that we're analyzing is sort of the existing data that is in the EDW, and having a finer granularity on the genomic background will give us the most precise way of estimating the risk for with that individual without having then to state anything about their ethnicity per se.
Right. Okay. I have another one. I'm just going to my questions there. But have you had any surprises where you thought something would be a risk factor, but it turned out to be protective?
We haven't seen that just yet, but this is really in the early days. For this paper that we're presenting, we need to convince the reviewers that this is valid. And so we're really going after known relationships, like between sex and congenital heart disease, things that a cardiologist would acknowledge, like the risk of transplant when you're on these various medications and things like that. So that's really the first place. But I do think it's always been described that the maternal health has an impact on the child. And this is really quantifying that effect as far as the risk for congenital heart disease if the mother has pregnancy-induced hypertension. and also then highlighting what we generally know, that if you have congenital heart disease, you may have other systems disordered as well. Well, this is telling us that an 11-fold increased risk of having a neurological disorder in the setting of congenital heart disease and pregnancy-induced pulmonary hypertension, or pregnancy-induced hypertension.
Yeah, it seems like the current state of health is important, just like the genes, that your genome doesn't change over time. Speaking of that, have you been looking at the RNA transcriptional profiles, to change treatments? I saw something about PCAR Network doing that for febrile infant. I thought, well, maybe you do this in cardiology.
Yep. So the great thing is we can add any data set into that corpus of data for analysis. So the PCGC consortium has RNA sequencing data from the myocardium of patients with congenital heart disease obtained at surgery. And so that data can get built in, and we plan on doing that within the next several years to incorporate the expression data from the heart in patients that carry a mutation or don't carry a mutation. And then we have the outcomes from the postoperative surgical procedures.
Oh, that's great. And then my last question is, one day I think this will be available direct to consumer. People will just order their 30 million exome, thing and come to the pediatrician and ask us to interpret that. And what do we do?
Yeah, I hope that it all occurs within the context of the healthcare system. So I would hope that healthcare insurance providers would find value in their subscribers having sequenced a genome, so that information could be incorporated into the medical record, so that it could be used in a more preventative way, and then ultimately lower costs over the lifespan of that patient. So that is a way that then the data comes into the system in an orderly way. The variants are there. We could design our software tools to compute on that data and make it available to physicians. And I'll give you an example of one of our rapid whole genome sequencing patients in the CICU, a patient with congenital heart disease. That genome was sent to Rady. The child had a heterotaxy type syndrome, complex cyanotic heart disease, and we didn't find any variants that explain that phenotype. But the patient then went on to have multiple thromboses, ended up thrombosing the BT shunt, and almost died. And so went for emergent surgery, had the shunt revised, and then we sent information back to Rady saying, hey, this kid is clotting. Well, that wasn't part of our original phenotype description, so they didn't give us any information. It was there. We didn't ask for it. And then they say, oh yeah, this patient does have a variant in a gene that promotes thrombosis. So the better way to do this is to have access to all that data from the very beginning, so that we would be heads up, this patient is at risk for thrombosis, and maybe we could have done something differently and not have the patient, nearly die.
Right, and it seems so important on a patient with different heart disease and sick problems. And you think about when this opens up to general population of well people kind of seeing, oh, I have a bigger risk for this or that, there just are not going to be enough genetic counselors to handle all the interpretations. So the tools that you're coming up with really I think are going to be so helpful for everybody. And then the way you are doing this prospectively to kind of help people ahead of time, that's just awesome. I love it. Thanks so much for teaching us this morning, and thank you to everybody for joining us.
Thank you.
And we'll see you next Thursday. Thanks again. Bye-bye.